Hey everyone, and welcome back to the Ontario Aquaculture Research Centre. I'm your host, Kaylee Moore, one of the agricultural assistants here at OARC. If you're looking to learn more about aquaculture, you're in the right place. On today's episode, we have Dr. Nick Bernier, University of Guelph professor, joining us to talk about his current research.
Hi guys, and welcome back. I'm super excited to introduce Dr. Nick Bernier. He's a University of Guelph professor. He's currently doing research with us, so I'll let him introduce himself for you.
So hi everyone. I'm a professor in the Department of Integrative Biology at the University of Guelph. I've been there for about 20 years now. I run a lab that essentially studies the physiology of fish. We do a number of different kind of basic research projects, but also applied research projects, and some of that applied research is carried right here at Alma.
Yeah, thank you. So we were just talking about you being a professor at the University of Guelph. You run the department, not run the department, but you're a professor of the department in the integrative biology, and your focus is on understanding the pathways by which the environmental stressors are perceived and processed. Can you kind of put that in simpler terms for us?
Sure. Yeah, that's a bit of a mouthful. I guess it's meant to be concise on our website, but let me explain. In a nutshell, essentially my students and I are interested in figuring out how environmental stressors impact the biology of fish. The types of environmental stressors that we've been studying include things like hypoxia or lack of oxygen in the water, high levels of ammonia. Both like high levels of ammonia and hypoxia are things that you can see in high intensive aquaculture, but also in polluted waters, for example. We've looked at the impact of elevated temperature. We've also looked at what happens when anadromous or salmonids go from freshwater to saltwater environment. Essentially, when I talk about the mechanisms that enable fish to recognize those stressors and the impact of those stressors, well, we're interested in figuring out basically what are some of the natural adaptations that fish have to cope with these stressors, and then what happens when the stressor is kind of too intense or too long and they can't cope with it, and what are some of the negative impact on the health and the performance of fish. So, and we've essentially, we've carried out a number of research projects to try to address those questions.
Yeah, and we've been fortunate. I mean, we are currently in the works of 2 separate projects here at our facility. So after this, you've just been recently awarded funding for the GenFish project. Can you tell us a little bit about this project?
Sure, absolutely. So GenFish is a Genome Canada sponsored project. It's spearheaded by Dr. Daniel Heath at the University of Windsor. And Dr. Heath has essentially assembled a team of 25 researchers from across Canada, actually from 13 different universities. It includes a number of industry, it includes a number of government agencies, several First Nations, as well as several NGOs and Alma. So because a lot of the validation of some of the tools that will be developed through GenFish will take place here at Alma. And essentially GenFish has 2 goals. One of the goals is to develop a kind of a fish health toolkit. Now I'll explain what that means. And the other one is to develop a kind of a fish survey toolkit. So the fish health toolkit, the idea there is to use kind of modern molecular biology tools to look at the expression of like a whole panel of genes that regulate a variety of different physiological functions in fish. So whether it's immune functions, growth related, whether it's kind of the ability of fish to process different types of fuels, to deposit energy, to consume energy. So essentially the idea is to use non-lethal sampling on fish, whether it's in an aquaculture setting or wild fish in a natural setting, and by looking at the expression of a battery of different genes, be able to monitor on a continuous basis the health and performance of fish. So that's the fish health toolkit. The other toolkit is kind of the fish survey toolkit, and that's essentially developing, once again, molecular tools that can be used in the field to monitor, quantify kind of environmental DNA in order to figure out, if you take a water body, whether it's a stream or a lake, a freshwater environment, to figure out like what species do you have, what species of fish do you have in that water body, how many do you have of each species, by using environmental DNA to figure out, to analyze stomach content, figure out exactly what each fish are eating, and finally to figure out kind of what's the general food web of that water body. And you can do that using environmental DNA, but a lot of these tools still have to be validated and developed. So it's an ambitious project. It involves a whole bunch of people, but it has a lot of really practical applications.
No, that sounds like there'll be some very useful tools to use in the future. Like you were saying, like these all need to be validated and all that stuff, but I think that will be very handy moving forward for the aquaculture industry. So you currently here are doing a research project with us, and you are doing a non-lethal transcriptional profiling of growth in rainbow trout. What do you hope to learn from this project here?
Yeah, so basically have two goals. We've designed a couple of experiments that kind of use a very traditional called kind of fasting compensatory growth protocol. So what that means is that we fast fish for a certain period of time, and then when you start to feed them again, they go through the kind of what's called compensatory growth, and this is a really accelerated period of growth after a period of fasting. Fish are really good at doing this because they actually do this in nature, right? They don't feed in the winter, but then the spring comes, they have food available, and they have this really accelerated period of growth. But we can do this kind of in an aquaculture setting, and we can take advantage of this to basically look at what are the most important genes that are regulating this really fast period of growth. And so the idea, once again, is to, as we develop these molecular tools, we want to be able to test them in a fish culture setting, and we want to be able to essentially use non-lethal sampling of kind of muscle tissues, or in this case also gill tissues, to figure out whether, when we look at the expression of this whole panel of genes, can we get a sense of what are the most important biomarkers of rapid growth in an aquaculture setting? What are the most important biomarkers of fish health and fish performance? So that's one of the objectives of the work that we're currently doing here. The other objective is that we don't know really what's the health impact of doing kind of non-lethal biopsies of fish at a regular interval. So we've got a 3-month-long experiment right now at Alma where every 3 weeks we're taking essentially kind of little muscle plugs that are about 4 millimeters long, but about 1.5 millimeter in diameter. So these tiny little muscle plugs, but from these little muscle plugs that we take at a 3-week interval, we can measure the expression of 100 different genes in these fish, and we can track individual fish through time. So we can figure out, whether if you fast a fish for a long period of time, or if it goes through an accelerated period of growth, what's the profile of these different genes in the muscle, or for example, in the gills, important tissues for the health of the fish, and important tissues also just for, production. And we're trying to characterize whether this kind of repeated non-lethal sampling will have any kind of impact on the health and the performance of the animal. It sounds really simple, but those kinds of simple projects have not been done.
And they're important.
Well, they're important. I mean, they have applications.
Yeah. So you kind of talked about your one objective with aquaculture and this project, but how are these results going to be applied to the aquaculture industry?
Yeah. Well, I'll give you a very simple example. So let's say you want to do, you have a new feed and you want to do a nutrition trial. And you want to figure out, for example, you want to replace, fish meal with, let's say, insect meal. I think those kinds of research projects are very common these days, right? As we try to move away and replace some of the fish meal in the diets of salmonids. And you can do the traditional way, which is you can run your experiment and then on a regular interval, you can sacrifice a number of fish and do your typical biochemical analysis, and you can look at a variety of different performance index. Or you could just euthanize your fish at the end of the trial. So what I'm proposing is that maybe you could use some of these new molecular tools that we're using to track individual fish through time and repeat sample them by taking these tiny little gill or muscle biopsies on a regular interval to figure out not only what's the growth performance of these animals, how is it affecting, for example, lipid deposition in the muscle, how are these lipid incorporated into muscle mass, is it affecting the health performance of these fish, is it affecting the health resistance of these fish? And you can do this on individual fish through time. So you can figure out, for example, whether your diet is having a different impact at the beginning of the trial, a different impact in the middle of the trial or towards the end of the trial. And so in a nutshell, you're going to be able to, I think, greatly accelerate how quickly you can translate the impact of your research to an aquaculture setting, to a fish culture setting. So maybe we can have faster transfer of research projects into applications for the aquaculture industry.
Oh, perfect. I think that's all I have today. I don't know if you have anything to add before we sign off here, but I just wanted to thank you, Dr. Nick Bernier, for joining me today and taking time out of your research day. I know you come out here only every 3 weeks, so thanks for taking time to sit down with me.
Hey guys, I hope you liked the video today. If you did, please swim on over and hit the like button and subscribe. Comment below if you have any questions you want answered in any of our future videos. Hope to see you next time.